Thanks for coming back to our final session of the day. And we're going to have two more presentations. And the first one is from Laura Ranum at the University of Florida, and it's about myotonic dystrophy type 2.
Okay, thanks, Charles, and thanks for the invitation. It's really a pleasure to be here. I'm going to tell you about our new mouse model that we've developed for myotonic dystrophy type 2, and then try to draw some parallels across different diseases. So this slide you're familiar with, the top part, myotonic dystrophy, is a multisystemic disease. Just to remind people, the DM1 repeat is a CTG. You don't need any reminding of that probably, but the DM2 is a CCTG, so very similar repeat motif. The CCTG expansion mutation can be quite a bit longer and go up to 11,000 repeats, and probably even longer than that. There's a lot of somatic heterogeneity. And so this is a southern blot showing the DM2 repeat lengths in human DNA, and the smeariness represents the somatic instability. So the repeat length is different in different cells in the body, and that's from the blood. The expanded allele can be a discrete band, one or two discrete bands, but it can also just appear as a smear. And it's really hard to size these repeat expansions. This is an example of monozygotic twins that were collected a long time ago when I was working with John Day, and they're you can see that they have a repeat lengths that are quite different in size. This is a repeat expansion from the same individual that was taken first at 28 years of age and then later at 31. So the repeat length is growing over time. So the disease mechanisms in myotonic dystrophy type 2, when we first cloned the gene, thought this is just an example of an RNA gain of function effect. And there are RNA foci, there are splicing changes. And so that is true, but it's probably not the whole story. And those studies initially focused on the sense CCUG expansion transcript, but we found some years later in a different disease that there are also RAN proteins that are made. And in myotonic dystrophy type 2, those are leucine, proline, alanine, cysteine repetitive proteins that make that repeat motif in all three reading frames. There's also an antisense transcript, and that antisense CAGG transcript makes QAGR tetrapeptide proteins in all reading frames. So these are called repeat-associated non-AUG proteins because they could be made without an AUG initiation codon. And they have been found in 11 different diseases. So in addition to DM2 and DM1, they are found in a bunch of other disorders, and they can be homopolymeric, dipeptides, tetrapeptides, pentapeptides. There are sense and antisense RAN proteins in DM1 and DM2 autopsy brains. And this is just an example of some of the aggregates that you can see in red. There are also sense and antisense RAN proteins in regions of the brain that have pathological changes. And so this is DM1 showing the polyleucine sense RAN protein, a polyserine antisense RAN protein, and in regions of the white matter with these RAN proteins, where these RAN proteins accumulate, there's evidence for demyelination. So if you stain with Luxel Fast Blue, you get a demyelination. These areas also contain reactive astrocytes or neuroinflammation and microglia. So there have been quite a few DM1 mouse models that have been made, but there are no DM2 mouse models that are available. And that's a problem. If we had a good DM2 mouse model, then we could use it to better understand the similarities and differences between DM1 and DM2, but also across multiple repeat expansion diseases. And we thought that we've tried to make this model for a whole bunch of years. This wasn't the first attempt. We tried to make a cDNA model overexpressing the CCTG expansion. We did a whole bunch of other things. And finally, we settled on an approach that would use a bacterial artificial chromosome, because we had some success with that for SCA8 and C9orf72. And so a bacterial artificial chromosome is just a large piece of human DNA cloned into a bacterial gene that then you can use to make a mouse. And the advantages would be that it contains the full-length human CMBP gene, substantial flanking sequences, so it will be using the human promoters. And given that this disease gene is expressed in such a complicated way with both sense and antisense transcripts, we thought that that was probably a good idea. So we took a human patient cell line and used that to make a bacterial artificial chromosome library, and then screened that library for the CMBP gene. We had 3 clones. The 2 shown in those blots, one had the normal allele and one had an expanded allele. And the problem with cloning a big repeat that's unstable, if you have to go through E. coli, is that it contracts. And so we developed a strategy to try to prevent that, at least to prevent it enough to get our mouse made. And the strategy was to plate the cells on plates and grow the E. coli there, so that the faster growing contracted allele couldn't take over the culture. And these are 11 different cultures of the from 5 different plates each of the expanded allele, where we just tried to grow it for a very short amount of time and then get the expansion. And we ended up with a prep that we liked the size of. It was about 750 repeats. And then we did pronuclear injection to generate some transgenic mice. And this is Avery Engelbrecht, an MDF scholar and graduate student. And he started working on the project about the time when we had some potential founders. And he identified 2 founder mice that contained the gene. And from those mice, he's established 2 different lines. Their 236 line and the 75 line. And he's characterized the gene that landed. So the top shows the back as we understood its structure. And then once it landed into the mouse genome, the 236 line seems to have all of the parts of that back with the 5' and the 3' flanking sequence, but the 75 line has the entire CMBP gene and some 3 kb beyond the 3' end, but it doesn't contain this downstream gene. And that turns out to be a nice control, because there is a downstream flanking gene that if there's a difference in the phenotypes, we can control for that plus integration site differences. So RT-PCR shows that the human CMBP RNA is expressed in muscle in both lines, and it's also expressed in the brain. There, by generation N2, we already detected some repeat instability, which we were excited about. So there's this N1 mouse had about the same size repeat as the founder, but you could see little creeps up and down. And we were particularly interested in those sizes that crept up, because from from knowing about the mouse models of myotonic dystrophy, it seems really important to get the longest possible repeat that you can. So we started to do this selective breeding strategy to generate animals with longer repeats. So the progeny that had a little bit longer repeats, then they were bred to generate animals with longer and longer repeats. And now by N6, we're up to 1,950 repeats. So we're really excited about that, because most of the characterization that I'm going to talk about is with mice that are in the 750 to 800 repeat range, but we don't have very many animals at the about 2,000 range, but we hope to get a bigger colony of those mice with bigger repeats very soon, and we hope to keep growing the repeat. So intergenerational expansions are really frequent, with the male transmissions showing an even larger increase in the 236 line. But there's a lot of increases overall. We can do the genotyping by long-range PCR, which is really, by just PCR, but we can go across the repeat, which is important to make sure that we're controlling for repeat length in various studies. And we can do that with a long-range PCR that crosses the repeat expansion, and we can get across quite large repeats. And this is an experiment done by Avery that looked at DNA from different tissues, and there's and including the initial tail biopsy and then the final tail. And you can see that there is somatic instability, and the DNA repeat length grows over time. And there's also really striking repeat instability in the testis and the liver. And this is a PCR-based assay. So Avery's following this up with Southern-based assays to make sure that he's getting the whole picture of the repeat instability. But clearly there is somatic instability, and it seems to be quite variable in different tissues. And so when he does small pool PCR, he sees repeat instability that looks like it did in the previous slide. And this will allow him to get more precise estimates of just how variable the repeat length is in different tissues. But it seems to be largest right now in the liver and testis, and less with less repeat instability in the brain so far. So does our mouse model have any molecular phenotypes? Avery has really been very persistent in trying to get at this question, and has used HCR fish to show that the CCUG RNA foci accumulate in both the 75 and the 236 lines in the skeletal muscle. He's also shown that these RNA foci accumulate in the brains of both in the frontal cortex, the hippocampus, but throughout the brain in both the DM275 line and the 236 line. In collaboration with postdoc Lisa Romano, Lisa has done some immunohistochemical staining to try to detect RAN protein aggregates, and she's found evidence for LPAC aggregates in both the 75 line and the 236 line, and they're particularly abundant in the corpus callosum, as shown here. So when I hear these beautiful presentations about targeting the sense transcripts, I just want to remind those who are in the industry to not forget about the antisense transcripts, because the antisense transcripts are producing another set of RAN proteins, the QAGR RAN proteins, and those are also found in our mice. So that's what's found in the human, that's what's found in the mice. In the regions where LPAC is abundant in the white matter, we see evidence for white matter abnormalities, or with the Luxel-Fast blue staining showing demyelination. So in the summary of this part of the talk, I want to connect these mice to another story. We have two independent lines of transgenic mice. They have substantial human flanking sequence, which means that they're using the human promoters to drive sense and antisense transcripts. These RNAs are expressed in the skeletal muscle and the brain. There are RNA foci in skeletal muscle and brain. There are sense LPAC and antisense QAGR protein aggregates that accumulate in multiple brain regions. And the CCTG repeats are intergenerationally and somatically very unstable. And we're excited about this because we think that time will improve our model, we're hoping. So the some of the future directions are, we need to do a lot more work. So these are all unpublished, very recent results. We need more phenotypic characterization of the animals. We need to really understand the skeletal muscle and the brain histological features. So we're going to focus on that initially. We need to but we think it's a great tool to really understand the molecular mechanisms of this disease. So is DM2 driven primarily by an RNA gain of function mechanism? Is it driven by RAN proteins? Is it driven by a combination of the two of them? With this model, we'll be able to tease that apart. What's the role of the sense transcript? What's the role of the antisense transcript? If you target the sense transcript, to elevate the antisense transcript, which is something I worry about with the ASO trials. So we could also use this model to try to understand the repeat instability and identify cis and transacting factors, and whether they're similar to some of the cis and the transacting factors that have been identified in Huntington's disease, and then for therapeutic testing. And this is where I want to segue to a different topic, and then I'll bring it back, hopefully. So RAN proteins have been reported in 11 repeat expansion disorders, but there's a lot of unpublished data out there that this is much more common than we previously imagined. And we've developed a similar back transgenic mouse model of C9orf72 ALS FTD. It, again, expresses sense and antisense transcripts and sense and antisense RAN proteins. And in that disease, we wanted to develop a way, well, in all of these diseases, we wanted to develop a way to turn off RAN protein production. We thought if we could just turn off the faucet of the production of the RAN proteins, then maybe we could really improve protein homeostasis and the health of these mice and maybe make an impact in the disease. And so that really requires that we understand the mechanisms of RAN translation better, or at least what triggers it. And so this is Tao Zhu's work, and he has been discovered RAN translation in SCA8 in the lab in 2011, and since then has really been working hard on trying to understand mechanistically how this works. And so what he's discovered is that expansion RNAs can look like viruses. They can activate the PKR pathway, and that upregulates RAN translation. And that work dovetails nicely with work of others that show the integrated stress response increases RAN translation. But we think PKR is a critical pathway, because when you can inhibit PKR with a dominant negative form of PKR, or we discovered with an FDA-approved, really cheap drug, metformin. And that reduces, so I'm going to show you some example data from the metformin study. It reduces RAN protein levels in cells, and that's across multiple types of repeats. It reduces RAN protein levels in C9 mice, and that's from the metformin study. It decreases neuroinflammation. It improves behavior, digigate and open field behavior, and importantly, it improves motor neuron survival. So that's a classic feature of ALS and frontotemporal dementia, or ALS, is motor neuron loss. We see that in our mouse model, and then that's rescued with metformin treatment. And blocking the PKR pathway with metformin does not change the levels of the RNAs or the RNA foci. And so metformin is really in wide use. It has been in worldwide use for decades. It's really cheap. It's been shown to have beneficial effects in a whole bunch of mouse models: Fragile X mice, myotonic dystrophy mice, or myotonic dystrophy patients, Huntington's disease patients, and we are now testing it in an initial trial to see if it reduces RAN protein levels in C9orf72 ALS FTD patients. It's been used in Huntington's disease. It's been shown to work in 3 different mouse models of Huntington's disease and in patients. So I was really inspired this morning by Andy's talk. I don't know where Andy is, but and just the idea that maybe, maybe we can make an impact in these diseases with cheap drugs, but also with natural compounds. And so we've been testing a natural compound, and we approached it with the lens that this natural compound can reduce RAN protein. So that's what we were looking for. And this one works at a micromolar level. So it's substantially more effective potentially than metformin, which works at a millimolar level. But so we're excited about that. We've used this compound to test gait in C9 or 72 ALS mice. So we've just completed this big trial using this compound in the mice. And you can see these differences in digigate behavior between the non-transgenic and the C9 mice, and then the treatment with this drug rescues those changes. And altogether, there are 13 different digigate abnormalities that are found between the C9 versus FVB control mice, and all 13 are corrected by this compound C. So finally, I just want to thank you for your attention, and especially acknowledge Avery Engelbrick's efforts. So his poster is number 10. It's, as you go through those side doors, it's the first one to your right. He's got many more details on his poster than I've been able to cover. And also Lisa Romano's been a big help with this study and the characterization of these mice. And then there have been two fellows that have been supported by Myotonic Dystrophy Foundation for this work. And so Kiru and Avery, and it's been a hard project. So it's taken a lot of years to generate a DM2 mouse, but we're really excited about the one we have, and we hope that it will really help our understanding of myotonic dystrophy type 2 and allow the development of new drugs. So thank you for your attention. I'm happy to answer any questions.
Hi, Laura.
Hi.
I'm super excited about the DM2 mouse. Have you guys looked at splicing phenotypes or any like functional deficits or anything yet? I know it's early. But...
Yeah, so Avery's looked at that, and it doesn't find splicing changes right now, but he's looked at mice with about 750 to 800 repeats, and at a couple of different time points. So some young, some old. I think the assays are working beautifully, but I don't think there are splicing changes yet. So we may need to grow this repeat.
Hopefully
further.
with the repeats.
Yeah.
Awesome.
Hi, what is the expression level of this transgene in relation to the endogenous gene?
We don't really know that yet.
Still working on that.
Okay.
There's some extreme similarities.
Got it.
Yeah, so the northern blot, it's just this transcript runs at this exact same spot. So we have to make some human allele specific.
What about
probes.
at the RNA level?
Well, at the RNA level, the transcripts identical size and very abundant, and so we're not sure if it's what is due to the transgene and what's not. I mean, I think it is. We need to do some more quantification. I think it is showing a band by the northern that's significantly increasing the intensity of the band, but we need to do more to quantitate it.
Your metformin data is quite interesting. As you know, metformin is a very dirty drug, just like exercise is dirty, and they have a lot of similarities. So, as you, I'm sure, are aware, metformin can inhibit complex 1. It can also reduce the activation of the NLRP3 inflammasome activation, which may be part of the reason why you're decreasing the inflammation. So it's a very interesting drug, and it has either AMP kinase dependent and independent mechanism. So it's really interesting. It's very similar to what exercise does.
Thanks.
Just a comment. It's interesting, yeah, but not unexpected. And a lot of those effects you've probably seen, we and others have done exercise training in the G93A mouse model,
Yeah.
showing some similar benefits. Yeah.
Now the metformin doesn't work in the G93A mouse model, but exercise does. So metformin doesn't work, and we think that's a pretty good control actually for our studies, and maybe an indication that targeting the RAN proteins improves things. And I didn't think I had time to say this. Maybe I had a lot more time than I thought I did, but we have done another complete study where we've used antibodies to target the RAN proteins, and we get similar results where we improve the motor neuron survival. We improve the survival of the mice in that study, and we don't change the RNA, but we do change the RAN proteins.
Do both lines show the same antisense expression, since the other line is truncated towards the 3' end? And do you know where the transcription initiation is for the antisense?
Don't know where the transcription initiation begins yet, and we haven't even looked at the antisense RNA yet. We've looked at the antisense QAGR proteins, and that's not there's no way to compare yet. But in DM2 patients, one of the things that John Cleary learned when he studied the DM2 transcript is that the antisense transcripts go way up in the DM patients, in the DM2 patients, and that also occurs in C9orf72. So there's something about the disease process that's triggering that antisense elevation.
Laura, from the HCR fish, the signals for example, in the brain, they're very bright.
Yeah.
From both strains.
Yeah.
Yeah.
But the HCR fish is much more sensitive than regular fish. So it could be that we just haven't yet looked at the right DM2 specific splicing changes, or that RB Fox is going to be a player here. But because we just expect to see some more splicing changes if they were really abundant. And so Avery can talk more about that at his poster, but I think we're not sure that they're big foci. He's really good at detecting the foci.
Okay, so the reason I asked was, do they seem confined to the nucleus, or can you see them in the cytoplasm and
I would say we haven't looked good well enough.
I'm sorry.
Can you find them in the cytoplasm?
The foci? I wouldn't say so. No.
Paranuclear, but not really in the cytoplasm.
But some RNAs.
Isn't that in the cytoplasm?
Yeah.
Not.
I don't know. And do you know do you know about intron retention or how?
No, and I think that's an excellent question, because if you there's a big hot topic in the Huntington's field about even there could be we do know, I guess, from the olden days about intron retention that there is splicing, but we don't know if there's and we do know from Lukash's study that was published in Maurice Swanson's lab that there is intron retention in DM2. We don't know that in the mice yet, but that could be, that could also have parallels to other diseases, because in Huntington's disease, they think that there's just a truncated form. The repeat is in an exon, but they think that there's a truncated transcript that has a different poly A site in intron 1. So you get a short transcript, and then you can also get the full transcript, mRNA. So I think there's a lot, we don't know about a lot of things, including these mice.
Any other questions? Thank you very much.
Okay, thank you.
Okay, and so finishing off today, we're going to have a presentation from Eric Wang about, there's a lot of conversation at this meeting about oligonucleotide therapies, but not so much about the gene therapy of late. So we wanted to try and have a presentation on that. Thanks, sir.
Great. Well, thank you, Charles, and thanks for your attention. The task that I think I was given was to talk about gene therapy broadly, and so a lot of the work I'm going to talk about has nothing to do with my lab. It really is observations from other work from other labs, other work in other fields besides myotonic dystrophy as well. So just my disclosures. And then just a brief outline of the things that I'm going to try to cover, the topics I'll cover, just some basics of AAV biology, which are quite interesting from a basic perspective. An overview of the various gene therapy approaches that have been taken for DM. And then I want to highlight a number of the questions and challenges that face gene therapy as a modality in the context of myotonic dystrophy in particular. So when when the word gene therapy is used in the field, it can mean a lot of different things. But for the purposes of this presentation, it's really going to focus on viral mediated delivery of exogenous cargos, and primarily through the use of adeno-associated virus. So adeno-associated virus was discovered quite a long time ago. It was discovered as a contaminant of adenoviral preparations back in 1965. And it requires adenovirus genes to be present in order to replicate. And that's where it gets its name. And it's a very widespread, prevalent virus. Over 50% of individuals, it depends on the publication you look at, but quite a few individuals are seropositive for AAV2, but there are a lot of different kinds of naturally occurring variants. Basically, the capsid sequence is really what defines that. But it's quite benign. It contains it's made of a single-stranded DNA virus that's about 5 kilobases long. And in the natural form, it contains 2 specific genes, rep and cap. And these are processed in many different ways. They're alternatively spliced. They use alternative translation. Many of us are now very familiar with virus biology from the past couple of years, and how viruses have very unique, compact ways of using gene expression machinery to do pretty amazing things. The major part of AAV that is going to be really relevant for the work that's ongoing now in the field is really in this cap protein, this capsid protein, which comprises a single sequence, but can be made into 3 different open reading frames, the VP1, 2, and 3 protein capsids. As you can see, there's overlapping open reading frames. It's quite complicated. The virus biology of AAV is complicated, but early on, Jude Samolsky, Nick Mazishka, and others at the University of Florida, interestingly, I serendipitously inherited my lab space from Nick Mazishka, so I was forced to learn about AAV biology, and maybe that's why I'm giving this talk today. But so the production of AAV to carry exogenous cargos, essentially you take the rep cap out and then you provide it in trans. So rep cap is provided in trans, but you can put your own cargo that you wish to be packaged into the AAV between those ITRs, those uninverted terminal repeats. And again, because AAV requires adenovirus to replicate and be packaged properly, you also provide some of those components in trans. And there's lots of different ways to do this. There can be a transfection-based systems. There can be stable cell lines that express this. There can be other organisms, cells from other organisms that can produce. But in essence, the material between the ITRs and the ITRs themselves get packaged into a particle. And this particle is made up of a 60 subunit capsid of VP1, 2, and 3 that captures that whole genome inside. And there are different surfaces on the capsid that have been the subject of a lot of engineering in the past years. One of the really hot locations is that 3-fold axis, that interface between the VP proteins that in which people have been able to add short peptide sequences to give the virus new behaviors, new tropisms, affinity for different receptors, different abilities to enter different cells. And so that's a little bit about AAV. So what about what gene therapy approaches for DM have been tested? Now, one of the attractive parts of gene therapy is that many drug treatments out there, small molecules, traditional medicines out there, require you to go through some sort of process that is semi-rational or not so rational, right? You do screening. And what's exciting about gene therapy is we can take all of the knowledge and expertise we have in molecular biology, and we can start to become a little bit more rational about what kinds of behaviors we want. And so for that reason, the ways in which we can manipulate biology span all aspects of biology, from DNA to RNA to protein. And so at the level of DNA, there are recent efforts to try to remove or replace the expanded repeat tracks. Primarily, the work has been done in DM1, but with new models, we'll be able to try this in DM2. There is interesting literature from Vince Dion's lab in which approaches have been used to shorten the expanded repeat track. Our lab and others have used methods to impede transcription through expanded repeats. There's approaches using synthetic proteins to impede transcriptional initiation. Also, there's recent approaches and a number of companies that are pursuing epigenetic modifications that could allow for chromatin level or regional level silencing of certain loci. On the RNA level, there's a lot of approaches that have been taken as well that are complementary to ASO or siRNA-based approaches. Primarily, these have been focused on degrading a repeat-containing RNA. There's also approaches that include displacing RNA binding proteins from the repeat-containing RNA. And then finally, at the level of protein, there have been efforts to deliver exogenously additional protein that can try to compensate for the muscle-blind protein that is sequestered on these repeats. And so I'll go through each of these areas and try to provide some commentary on these different approaches and examples. And there are references here, but by no means are they fully comprehensive. They sort of represent examples of each of these approaches. So, as I mentioned, there's a number of approaches using CRISPR or Cas-based systems. Zinc fingers and talons as well, you could imagine, be applied to this system. There are caveats that have been highlighted as well that include the consequences of making double-strand breaks in the genome. There can be loss of uncontrolled loss of genomic regions. There can be other things that happen that are undesirable, but a lot of attention is being put in this area. An example from a relatively recent publication now uses Cas9 using guide RNAs that flank the repeats to try to delete that repeat region in DMPK. And RT-PCR can be PCR on genomic DNA can be used to assess the efficiency of removal of the repeat. Those lower bands represent instances, alleles in which you have removed the repeat and repaired to remove that sequence. When you select for cells that have undergone that editing event and look at things like RNA foci, you can see that these specific clones show less foci as expected. And you can also see rescue of missplicing in specific clones that you later assay and look at muscle-blind dependent splicing events. So these sorts of events are being pursued, many in cell-based models. Some have been taken forward into animal models. But in general, there is work to be done to improve the efficiency and improve the safety of this approach. There's a lot of attention as well being placed on RNA targeting approaches. And this history goes back quite a long time. Once there was a good understanding that the RNA species itself is toxic. Lots of different molecular biology tools and tricks can be applied to manipulating RNA abundance, all the way from ribozymes. Jack Poimaret has done this work very, very early on, 2003, also gone on to show sensitivity of toxic RNA repeats to shRNAs. Joel Chamberlain's lab has pursued microRNAs that target repeat-containing transcripts by adeno-associated virus, and a number of other clever techniques using U7 snRNAs. These are typically important for cleavage of histone ends and 3' end processing of histones, but they've been repurposed to regulate splicing or displace RNA binding proteins from other RNAs. That's been pursued by Denise Furling's lab. And then a number of other Cas-based approaches, deactivated Cas9 fused to endonucleases, familial proteins, these are RNA binding proteins that can be mutated to bind to specific sequence, CG repeats, for example, fused to endonucleases, and more recently, RNA, bona fide RNA targeting Cas proteins. I just wanted to highlight the early work. This 2003 paper was, I remember I was, this was before I started graduate school, but not that long before I started graduate school. And one of the exciting parts of reading the literature to think about ways in which we could get rid of RNA. And this approach really uses this RNA enzyme essentially that can fold into a specific structure and catalyze cleavage of a targeted RNA. And it was repurposed to target the DMPK RNA. And in these studies, you could see reduction in RNA foci. I'll highlight another set of experiments pursued by Joel Chamberlain's lab, in which a microRNA targeting the human skeletal actin transgene was delivered into mice by AAV, and they were able to see a nice knockdown of human ACTA1 and associated rescue in specific splicing events. And this was in the context of AAV6. So finally, protein replacement as an approach was really first pursued by Maury Swanson and others in this audience to not necessarily to provide a therapeutic strategy, but back then it was really to prove the hypothesis that muscle-blind sequestration is an important pathway that is activated, that is operating in the disease. And so Raul Cornelia, a postdoc in Maury's lab, packaged muscle-blind 1 coding sequence into AAV and did an intramuscular injection and showed that you could rescue several different phenotypes. So what you can see here is the exogenous MBNL by Western blot in that middle lane of the Western blot. It runs a little bit higher due to that MIC tag. And then a redistribution of the muscle-blind in nuclei, going from something that's quite punctate, that is sequestered on the repeat foci, to something that's a little bit more diffuse and perhaps soluble. And they were able to see a nice myotonia rescue in the tibialis anterior when they assayed that. One of the issues that has been raised with this as a therapeutic approach is that muscle-blind overexpression can be toxic in certain contexts, in certain cell types. Jennifer Davis's lab has been looking at this in the context of the heart. Also Denny Furling encountered this phenomenon, and through removal of the C-terminal domain of muscle-blind was able to mitigate some of that toxicity. So that's sort of a summary of a lot of the different approaches that have been taken using gene therapy. Really, the power of gene therapy is that you have about 5,000 bases to work with. So you can choose whatever sequence you want to put into that virus. But what is it going to do once it goes into cells, right? So I'm listing here a number of questions and challenges, and I'll go through each of these briefly just to highlight not necessarily obstacles that are going to prevent this being usable in the myotonic dystrophy context, but things that we need to think about and things to optimize and things to develop in order for us to be able to effectively use this for this disease. So this issue of dose of AAV, there have been a number of clinical trials now being performed for other muscular dystrophies, including myotubular myopathy, spinal muscular atrophy, and Duchenne muscular dystrophy. And if you take a look at the doses that are used, you're in the E13 to E14 range. So 1 E14, that is 1 trillion viruses. You have about 37, that's 100 trillion viruses, and you have about 37 trillion cells in your body. So that's 3 viral genomes for every cell in your body. And using existing approaches, most of that goes to your liver. So these trials, while in some individuals have shown really dramatic beneficial effects, some of the kids and some of the people in this audience have been able to witness this firsthand. Some of these effects are extremely dramatic, but at the same time, there have been severe consequences, deaths occurring in response to high dose, my tubularin AAV as well as the mini dystrophin setting. And so one of the reasons and one of the mechanisms that has been proposed to mediate sensitivities to AAV is perhaps underlying liver disease, at least in the myotubular myopathy case. That's being proposed as one of the leading hypotheses of the issues. And so there is a lot of attention to try to better understand this. And in the context of XLMTM, this underlying hepatobiliary disease needs to be either addressed, or there needs to be ways to get around this. And so this raises the question of whether in the context of myotonic dystrophy, there is liver disease, and of what type. There are reports of abnormal liver function. I heard from someone that there was a discussion in one of the patient sessions about liver enzymes and liver functions, and there is some literature on this. There are some investigators looking at this, developing liver models of myotonic dystrophy, but I think this is something that is important to pay attention to. More so than understanding liver function, the ultimate goal needs to be to lower the dose of AAV that is provided to achieve potent and safe outcomes in patients. So there have been a flurry of articles and news reports about where the gene therapy field needs to go in order for the approach to be effective and safe. And yeah, in the long term, we need to shift our focus from how we can safely use high doses to how can we design the vector so we don't have to. And at the same time, there has been many scientists that are working on better versions of AAV capsids that are much more potent, that get into desired tissues of interest, that do not get into the liver, using high throughput technologies really fueled by deep sequencing. So on the left, this is a publication from the Broad Institute. Really on the very left, a schematic, a very simple schematic of the scheme that is being undertaken by many parties, essentially creating millions of variants simultaneously in a pool, and then administering that all at the same time in animals, in mice and non-human primates, and then pulling the material out, harvesting the tissue, and then using deep sequencing to understand which particular capsids were effective to get into the locations of interest. And so through this approach, the Broad Institute was able to identify specific capsid variants that get into muscle 10 to 50 times more efficiently than AAV9, and also get into the liver at the same or maybe half the levels in terms of total mRNA and protein production. On the right, you have a publication from Voyager Therapeutics, where similar approaches have been taken to alter capsid sequence and identify capsid variants that get into the brain very well, even just by an intravenous injection. And you can appreciate, compared to AAV9, you're seeing much more dramatic tissue tropism and effectiveness of expression in those variants. And so there's a lot of activity in this field. It's a highly competitive space, but in general for disease biology and therapeutic efforts, that's a really good thing, because we're going to end up with much better tools. And one of the things that I will note is that one of the phenomena that have been observed is that some of the variants that work efficiently in the screening system, so if you screen in one particular mouse line, they don't always work in a mouse line with a different background, for example. Or even if it works in mouse, it doesn't necessarily work in non-human primates. So the system in which you screen can be very important to, so you get what you select for, right? So that's something really important to keep in mind. And it does create challenges and bottlenecks in terms of really doing the work, because you need access to those appropriate systems. Moving on to another question and challenge: is there pre-existing immunity to AAV capsids? I mentioned that there exists pre-existing immunity to naturally occurring AAV capsids, and there have been studies looking at this different AAV naturally occurring variants, AAV2, 5, 6, 8, 10, across a population of hemophilia patients. It was just easy to get the material, and they looked at this. And then John Day has looked at seropositivity in SMA patients to look at AAV9. And you see these certain proportions. And so this is an essential part of the screening process to be in a clinical trial. And of course, there's a lot of work that is being undertaken to find ways around this, to be able to dose in individuals who are seropositive, or to be able to redose in an individual who's been given a therapy before, because once you dose currently, there's really no way to do it again. Another question that's really important for gene therapy, because it involves nucleic acid needing to go into a nucleus and express a cargo. It's different than a small molecule or an oligonucleotide that may distribute in a more even fashion throughout the tissue. What are the cell types and tissues that really need to be targeted? So for myotonic dystrophy, we know we want to target skeletal muscle, but do we need to think about satellite cells? There actually hasn't been that much work done in our field to understand the contribution of satellite cell biology to phenotypes. There's emerging literature on this in the senolytic angle, but this is something that's important to think about. In the heart, we think about cardiomyocytes, but half of the heart is made of cardiac fibroblasts as well, and there are other cell types as well. In the smooth muscle, we think about the muscle, but what about the enteric nervous system? Is that playing a role in disease? And in the central nervous system, what are the cell types that we need to target? What are the brain regions we need to target? We actually don't have very clear answers on these at the current time. And so that dictates, for example, the regulatory elements, the promoters, the sequences that you need to incorporate into your virus. Now, let's say you know the cell type to target or the tissue to target. What proportion of cells or nuclei need to receive the material? So both the DM1 and DM2 mutations exert gain of function effects. So, and we know that the offending targets are commonly in nuclei. And at least in muscle, the mynuclear domain theory of muscle, where each nucleus is responsible for gene expression locally, presents important considerations for this. And so one of the examples in which we first were alerted to this was work done by Belinda Pinto and Tombi Saxena in the lab. And Belinda is here, and she would like to talk to people who are actively developing medicines in an industry setting. So she's right there. This is a more efficient way. Instead of introducing her to you, she's right there. And her email is bpinto@ufl.edu. So Belinda and Tombi pursued this idea to block transcription of expanded repeats by Cas proteins. And so it worked in cell culture, it worked in myofibers ex vivo, but when we went in vivo with gene therapy, we saw that the expression of the Cas protein was patchy. You can appreciate here in green. It's not present in every nucleus. There are a lot of nuclei there in the myofiber that do not contain any Cas. We've done a number of other studies looking at RNA distribution in myofibers. And when you look at endogenous RNAs, they're pretty well dispersed throughout the muscle fiber. But when you go in with an AAV and look at an exogenous transgene, you can see that nucleus on the left, the one upper left and probably the one on the lower left, they're both functionally transduced. They're producing RNA. The RNA is coming out, but you have this whole other part of the fiber that is not transduced, and you don't have any RNA there. And so AAV transduction is not 100% efficient. So how many mynuclei must be treated? This is an important question to answer. And then in the context of cells with single nuclei in the brain, for example, how many cells do you have to treat in order to get a therapeutic rescue? How immunogenic is the cargo? This is quite important. We're already seeing this in the context of dystrophin gene therapies, where certain patients with certain mutations seem to be showing cardiomyocytis and not other patients with certain mutations. So there is the idea about immunity against certain sequences. RNA approaches have some advantages over this because in general, immune reactivity tends to be biased towards proteins. Long-term expression of foreign cargoes is problematic, and then pre-existing immunity can exist against some of these CRISPR enzymes. So that's very important to consider. Last few questions I'm not really going to spend much time on, but of course they're important in any medicine. What are the off targets of the cargo? This is particularly important for the CRISPR enzymes. A risk that is raised that I think the regulators are still forming an opinion about this is, what are the risks of AAV integration? It is not a frequent phenomenon, but it does occur. And then ultimately, because gene therapy is not 100% efficient, how do these factors impact how you measure therapeutic rescue? So I'll just sort of close to the point is not to dampen enthusiasm in these areas, but to raise the obstacles early and upfront so that we prepare and find ways around these obstacles. There's a lot of parties in this space that are actively pursuing gene therapy. And I think it's important that we're all aware of these issues so that we deal with them and don't get any surprises when we actually move forward with these approaches. A lot of these questions are surmountable, and it just takes time and effort. And I think the basic biology side to understand how viruses infect cells and how exogenous cargoes behave, I think that's really, really relevant in the context of gene therapy. So, thanks to a lot of the people in the lab who contribute a little bit of the data here. There are a couple other individuals in the lab I wanted to highlight. Emma Shea is a graduate student. She has a poster that is in the other room focused on peptide oligonucleotide therapies for myotonic dystrophy type 1. Also, Carmen Valero, Juan Arboleda are both here, very focused on CNS issues in myotonic dystrophy. And so I'll just pause there and take questions.
Just a quick, rather naive question. Do you know if people who have antibodies to AAV2 will have cross-reactivity to AAV9?
I'm not the right person to give you a solid answer on that, but I think there could be antibodies that recognize shared regions and antibodies that recognize unique regions. As you can see from the assessment of seropositivity across different AAV variants, there can be differences in,
Okay, I thought as much. Thanks.
So another naive question. I mean, is that a no-go? So if a person is positive for AAV, then you're not going to treat them with AAV.
I think that's generally the conservative approach right now, right?
John? There are strategies.
Yeah. Plasmapheresis, for example, these are, yeah. Yeah. Yeah.
I mean, the other issue is that for the most part, we're measuring binding antibody, but some companies are just using neutralizing antibody titers to determine whether or not to go forward. So that's a very different issue. The neutralizing antibodies are going to basically impede efficacy, but the binding antibodies might be more associated with toxicity. And so they're on different sides of that, and there are trials going with both, so that the FDA is allowing both approaches to go forward, and we'll see.
Any other questions? That was a fabulous review of this topic. Thank you so much.